So here we were getting the results of counting the words in War and Peace. So I thought, let's take a variant of this. And in a way what I'm trying to do is take you from what you're familiar with Hadoop and MapReduce and say, well, this is how you do a MapReduce type problem in Condor. It can certainly do it. And it's okay for data up to, I don't know, 10, 20, 30 megabytes of data. That's okay, that's not too big. You certainly wouldn't want to do this with terabytes of data. You would not be wanting to transfer terabytes of data to each of the nodes. That would be a mess. And that's where Hadoop starts to really kick in and be more appropriate. So the problem I'm going to set here is that we have some file of data, some DNA, and it's a few billion characters long, and we would like to count the frequency of lengths of, say in the first case, lengths of size 9. How often does, say for example, that particular pattern occur? Yeah, that's the challenge. So we take this as our data source and we say we'd like to find, in this, to start with lengths of size 9. So, because we're going to distribute this on the Condor cluster, or any cluster, this doesn't have to be, this isn't just for Condor, this is anything, we want to say, let's just only send to each node the chunk that it needs to process. Whereas Hadoop, the data is already there, isn't it? In your traditional approach, you know the data has to be sent to the node that's going to do the processing. So we split the data up using the same splitting we used before. So we've covered that on Monday. And you don't even have to use a Java code to do this. You can use Unix commands. There are default Unix commands to just split some of the file into a number of chunks. That's all this does. So it says split this file here, which is this, into 5 pieces. I'm not going to go through that code again, you'd be pleased to know. Then what we do is we set up a job that is going to run the code on a single sheet that I have just given you. And that code is going to look for these sequences of length 9 and count them all up. So to each node goes one of these 5 chunks, and it takes that chunk it's got and counts the sequences. Once we're done, the code will come back. We'll have 5 count files, 5 different files that say what are the sequence counts for the chunk that they got. And then we merge them back together. And we did that before with the word count. So that's the same code again. So the only thing that's changing is the job that goes off to Condor to say what are you going to do. It's such a familiar thing to do. I mean, this is what you're familiar with Hadoop, but Hadoop does the splitting for you, doesn't it? You don't even have to think about it. But okay, so Condor's more general purpose, so you have to do that bit yourself as well. Yeah?
If you split files, how do you account for that you're splitting exactly in a sequence?
You don't. You've got me. This is not perfect.
Yes, well, I mean, if you count it looking at a length that groups of 9, you're going to have one starting every letter, so you're definitely getting some split.
Yes. This is. I could have written more complex code to take care of what you say, to know that actually the split would take account of the length of chunk you want, and would always make sure there would be an overlap between the 2 segments. I can do that, but it makes the code look horrible. So for the purposes of demonstrating the idea and the understanding, you would fix that later. I haven't done that, but you've absolutely got me there. You have to take account of overlaps. In fact, this is, you're kind of drawing on something where Hadoop really doesn't work. Because Hadoop, you've got no control over the chunk. Yes? If you've got to do analysis of data that relies on contextual information of what lies either side of what you've read, or in fact, say at the beginning and the middle, you can't. You see the issue? Hadoop is no good for contextual processing at all. I can fix that with my Condor job. In fact, if I really wanted to, I could. My file's not that big. I could send the whole DNA chunk, data file to every single node, and just tell each one where to start and where to finish. And that would solve. That would get rid of the overlapping problem completely, and I would just have to make sure I told each one where they would begin and finish and got my indexing right. Because the size of the data is not an issue in this case. It's only half a megabyte or something like that, which is no big deal these days. Do you follow? Were you aware of where Hadoop falls down? Hmm? But if you need access to arbitrary positions all over the data file, which does happen, there's nothing you can do. You have to have a whole data file available to you. And that is often an issue, particularly with DNA sequencing. You actually want a lot of contextual information, and it's not necessarily the DNA that's right where you are. It could be 1,000 bases away from where you are. And if you don't have control over exactly which piece you've got, this is actually a very relevant question. I think I was going to mention it later, but you've got me now.
You could, if you know the sort of things you might look for, might look to others, you can send all that to the user.
Yeah. But you really have to start thinking about it. It becomes very, very tricky. I mean, if your file's massive, you've got no choice but to do what you say. If you have a terabyte of data and there's contextual information, then you're going to have to work your way around it. You can't use something like Conda because the data's too big. But this is an issue with distributed processing. Sometimes you do need data from around where you are, but not where you are. And at least here, I've got complete control of what gets sent and what comes back. You don't have control of how Hadoop decides to partition your data set. So that's the merge. You familiar with what, you got an idea what I'm going to do? Yeah. So that's the data again, as I said. Here's my Condor submission file. So this is the bit that's going to do the sequence processing. The only thing that's different is the bit in red. I have different arguments. So the first argument is which class to use, and I said use the class called sequences, which is this. I then tell that class here is the file I want you to process, which is going to be one of those 5 chunks. And then the 9 is I would like you to count sequences of length 9. Here I tell it what files to transfer. Well, if I'm going to process this file here, I need to transfer that file there over to the node. That's the only change from what we did before with the word count. Use a different class, here's the file, that's as before, and here's the sequence length. Everything else is the same general structure. If I wanted to do a sequence of 9 and then a sequence of 3, all I would have to do then is change that to a 3 and queue another 5 jobs. And I get sequences of 5 and sequences of 3 and sequences of 9. Can you see that once I've set that structure up, it becomes very easy for me to get arbitrary length sequences. And in fact, I'll show you it later. You can actually put this, you can put you can redefine the arguments and say Q5 and then redefine the arguments again and say Q another 5, all in the same Condor submission file. You don't even have to have multiple files. And I'll show you an example of that in a bit. So this is the code to do the sequence. We're about to look at it right now. To do the sequence counting, it's very, very similar to what I had before. We have the main where the whole thing starts off. We create ourselves one of these sequences objects. And then we call the count method of my sequences object. So in here you'll find right after main, there's a count method. It's about 20 lines down. We're about to come to it in the next page. And this gets passed in the file to process, that's that bit there, arg0, which came from here. So this will be seq-dna-0.txt or seq-dna-1.txt and so on. Yeah? That's where that's coming from. And the other thing that's getting passed in is the sequence length to count, and that's this value here. And we turn that into an integer. So this might be 9, it might be 3, it might be 7, it might be 21. Yeah. That's how it, I just want you to see how the information comes in. Any questions? There's some slightly doubtful faces. I know I'm not very sharp at 9 or 10. I'm unfortunately sort of get sharper as the day goes by and then can't sleep at night.
You say you're looking for a sequence.
Yep.
Do you know what that sequence is, or are you just looking for sequences that are repeating that length?
The latter. I'm just wanting to say, give me a count of all the sequences you find that have 9 places long. once, I'll count it.
But is that not just 9?
Just literally 9 bases. I'm just it's not a real problem in some sense. I'm just trying to say, well, we know that in pairs of 3 they make protein. So that's why I've got a multiple of 3. And sometimes proteins are made from sequences.
But we've got a string of just characters.
Yes, which correspond to the bases. Actually, I'm going to come to your question. How do I know what actually makes a protein? Is that?
Yeah, there is that. But then when I'm looking back, so given the whole string of characters,
Yeah.
and you've got 9 characters.
Yeah, and then the next line, and then the next line,
And
and
that
then.
happens as a rolling line.
Yes, it will do. It actually spans across. I'm going to cut. You're ahead of me, because I'll come to that. I'll show you how I'm actually going to do the counting, and it actually relates to the way DNA is translated into proteins, and it's mind-bending, to be quite honest. In a way. Frank? Well, it is indeed. It's amazing. I mean, one of the things I'd love to get into in research is understanding DNA as a programming language, because that's what it is. But it's you get the sort of simple science explanation of what's going on, and then you find out that's not the truth as usual, and the truth is so much worse. But in the same as impressive. But then you go, oh, this is going to be harder than I thought. Funnily enough. So I'll come back to you, if you can bear with me one slide or two, I'll come back to your question about exactly what I mean by sequence, but it's easy to show you the code. So here I say, here's the file to process, here's the length I want to look for. Now I start to do the counting. I'm going to use a hash map, just as I did before for the word counts. Instead of a string being a word, a string is going to be a sequence. And the count's the same. This is just word counting, really, in disguise. So here's my hash map. I'm going to create an output file for each of my sections. So if I was given a particular chunk to process here in file name, then I'm going to have a count file C, and this is going to be. I'm going to put in the file name the length of count it's for. So if it's for lengths of 9, that'll be a 9. If it's lengths of 3, that length will be 3. So it'll be, say, C-9- and then the psip processed. So it's the counts of length 9 for the bit that it got. Am I going too quick? Is it alright? I don't want to go too slow either, but I don't want to go too quick. So, right, so that's me creating file name. That I'm going to save stuff to. I'll come back to that at the end of this method. Now I open the input file that I've been given, the chunk that I've been given, and start looking for counts. Now I have, right, that's supposed to be an equals. I'm pretty sure, I have no idea how that's actually, yes, if you look in your code, that's an equals. It's not previous minus in.readline, it actually says previous equals in.readline, yes? For some reason it's got, I have no, oh, it is an equal. What? It is, it's a very narrow equals, and I need glasses, I think is the answer to that. I do need glasses actually, I've been in denial, but I think the truth is out. It's a now recorded truth. They are very close together though, aren't they? Oh, it's, yeah, from here it's fine, it's from here it's kind of blurred from the screen. Where were we? So I'm going to attempt at least to take a fact that sequences, if I'm reading this a line at a time, a sequence can cross over a line. So I'm going to take the previous line and then the next line, and always take that overlap so that I don't get fouled by the. I don't just go up to the end of one line and then pretend to start again. They're always like a sliding window of the previous line plus this line. So that's why I'm reading the previous line. In a sense, the first time I do this, the previous line is the first line. Then I read another line. And I hope that I've got at least 2 lines. And then I create a combined line, which is the previous line plus this line. Then I pass it to the magical method called count sequences, which we will come to. That's going to count the sequences in this combined line. and then I set the previous line to be the current line, and go back and get another line. And I keep doing this until I've got no more lines left. And obviously the magic all happens in count sequences. Yeah? But this is just grabbing 2 lines at a time and saying count the sequences in that, and they're all getting put into this hash map here. We're taking a count here. Yep, question?
In this case, the second time, you read twice everything that is in line?
So what was, I now go back through the previous line plus this line. Yes, I do, but there's a reason for this, which is, actually I might do, you might be right. I can't remember. Well, I'll show you what I do, but I think you might be right. I might be in trouble.
You should just have
The running line.
Don't read it.
Yeah, possibly. Okay, I might have to fix that. I'm trying to keep the code simple. Here I write out the contents of my hash map, having counted all the sequences. I am going to show you how I count sequences. So we just literally go, we open the output file, we decided what the file would be earlier on, it's C-9- this chunk. Now we go through our hash map, we get the key set, so the key set's going to be the 9 character sequences for our DNA. So we just get all the different 9 character sequences, and for each sequence we get, we say, right, what's the counter for that sequence? Right, write that sequence out, tab character, and what's the count? So, the bit that does the sequence counting. Right, so I have a strand now, which is 2 lines worth, and I have a size, which is say 9 or 3, which is the size of piece I want to try and make counts for. So what I say, I have an index into my current strand, and I set it at 0, and now I want to start pulling chunks out of here to see what they are. So if I've got an index, whatever my current index is, I need to make sure that I've got enough left in the strand to actually pull something out. So I'm saying index plus say 9. So when I start at 0 I'm fine, 0 plus 9, so if I've got at least 9 characters I'm good, but I might be near the end of the strand. You know, the strand might be 30 characters long, and I'm in the position, index is at 27, and I add 9 to it, and I haven't got enough. So I don't want to carry on trying to pull that sequence off the end of the line. So this is just to make sure that I'm always within, I've always got enough left in my strand to pull a given length out of it. So if I now know that, I then say, can you give me in the strand the substring that starts at position index and goes up to index plus size. So when we very start off, index is 0, size is 9, so I'm going to get the sequence of characters between position 0 and 9 not inclusive. Are you familiar with substring? So that gives me a sequence, and then I do exactly what I did before with the word count. So this is just a word effectively, isn't it? The sequence is just a word. And I say, I ask the map, do I have a counter already for that sequence? And if it comes back saying null, that's saying no you don't. Then I say, right, well put into my hash map a counter for this sequence, and we'll set that counter to 1 because I've not seen it before. Yeah?
If I had a sequence already and its value was null, what would happen?
You wouldn't, that shouldn't happen.
Shouldn't happen, but it could.
Could it? I don't think, in this situation it can't, because the only way a sequence gets into the map.
There's a method of hashmap to check if it exists.
Yes, but I know this. If a key exists, the count. If you can see, the only way things get put in for a given sequence is via this line here. Yeah, there's no other way. If I had been devious and gone put sequence and then null, then yes, I get your situation. But that, you can see in this code that can't happen.
Yeah.
So I know that if I get sequence, it'll have at least a count of 1 associated with it. I'm not putting, so I don't set up all the sequences first with nulls and then add them as I find them. I just literally create the count as 1 as I find them. So this is only the case where I haven't seen this sequence before. Then I say, right, well I've just seen it, so the count's 1. If I have seen it before, I just add 1 to my running count for it. This is exactly what we did for the word count. Are you okay so far?
English first class.
Ah.
One.
Yes.
So you are
Yes. And yes.
lying.
I'm glad you asked that. So you're saying, why don't I go by 9?
Yeah.
Well, two things. The sort of distribution I'll get out will depend entirely on where I start, if I was to do that. And strange as it seems, DNA is literally read like that. A given sequence of DNA can produce an entirely different set of proteins depending upon where it starts. And it can start at any particular amino acid. In fact, so DNA effectively is multi-coded. It's a bit like if you imagine you're writing Python code and you can generate a different output, but depending on which character you Python code you decide to start at. And they're all valid. Isn't that mad? So one sequence of DNA can generate lots of different proteins depending upon which base you start at. And that's perfectly valid. It's a very condensed piece of code. It also made me think, oh man, how to interpret this? Because it can mean anything. What's even more impressive is that it turns out DNA is not coded in one dimension as we see here, it's actually 3D. So what happens with the DNA is sections of it can twist round based on the current density of other chemicals in the soup, as it were, and just close off a loop of code, if you like to think. That's DNA's equivalent of if. It really is.
I know
It really is. It has a conditional test, which is basically if there's enough of this stuff in the soup, it'll cause the twist to occur and lock off the section. So that strand will not generate the proteins that it normally would. And only when the density of this chemical decreases does the strand unloop and open itself up and allow itself to be executed. It literally has a conditional test. And the while is just, while I've got the chemicals to do this, keep doing it. So DNA does use ifs and whiles. But the trouble with all of that is that if you took a strand of DNA and you're just sitting there looking at that text file, you can't work out what it's going to do because you have to get it to turn itself into its 3D representation and then model the twisting action to see if that section will get turned on or off or not. Yeah? There's so many, I'm really simplifying it still. So we can tell what, we can work out what base sequences might produce a protein, but we don't know if they will produce the protein under the circumstances in which it's operating. In a sense, the only way you know it's happened is you can see the protein, and you can see the sequence that caused the protein, and you know that that's how it's put together, but you couldn't take the current environment and work out what's going to happen because this, isn't it mad?
It's post-process.
Yes, and then it goes from...
genetic messenger or transfer.
Yeah.
And there's RNA processing of that before it gets a protein.
But it's
very complicated.
It's very complicated. That's why I'm just going to go off tangent here, but I find that, to me as a computer scientist, it's tantalizing because it's a programming language. Fundamentally, it's a programming language, and fundamentally, theoretically, we could work out what that language was. It really is. It's an amazing programming language, but it's devious. So I'll leave that with you, but it's just something that some days I think, wow, I'd really like to dig deep, and every time I look and go, wow, that's hard. So, but it wouldn't be science if it wasn't, you know. We've solved the easier stuff. We only get left with stuff that's hard, so that's the nature of it. Anyway, I better move on. So that's why we only go up one at a time, because literally those sequences, depending on which one you start at, could actually be interpreted. Right, so there you go. That was me running, I can't remember, I think the DNA sequence was of a fruit fly. Can't quite remember. I was quite surprised to find out that 9A's turns up 19,000 times in that sequence. I would have thought that would be pretty unlikely, but turns out it's... Yeah, it must be, but It is interesting, and you can see little things like this coming out. This was just done for fun. And I can run it again, by the way, with 3 by just running it without, instead of saying 9, say 3, and you get other variants. How am I doing? I'm so tempted to tell you stories about fruit flies and X-ray crystallography. I shouldn't, should I? Sorry. Some of the other work I used to do with genetic algorithms was to try and work out the structure of collagen. We know the amino acid sequence for collagen, but what we're not so sure about is exactly how the strands, the protein strands, line up. And collagen's an amazing structure. I mean, we would literally fall apart if it wasn't for collagen. So the way we were trying to figure this out was do what's called X-ray crystallography of collagen structure, and then we were trying to ask, then using a genetic algorithm to evolve collagen, the positions of the bases, to see if we could get our simulated X-ray crystallography to match up with the real crystallography. You follow? So we know what the sequence is, we don't know quite what the alignments are, so we got a GA to try and work out what the gaps between the amino acids must be, and if it chose a gap, it had to use that gap everywhere along the sequence of 1,024 bases. And then we would simulate, when we had that structure, I then had to write code to simulate doing an X-ray diffraction of that structure. And then I said, right, and what you said to the GA was, you're doing well if these match, and you're not doing well if they don't. And if you go to something else at any point, there's a poster on the wall somewhere shows the match we got, which was bang on. I was going to get to that, which is where the fruit fly comes in. So X-ray crystallography is firing X-rays, very high energy X-rays, through crystals in theory, and if you do it, if it's a crystal, you get a very defined pattern of where the beams go, because the crystal's a regular structure. So you can see if you shine light through a crystal, I'm diverting slightly, where the sort of light goes is very dependent on the crystal structure, and it does it in a regular fashion. Would you buy that? It's not quite true, and my analogy for it was it's a bit like firing a shotgun at a TV and then looking at where all the pellets ended up to work out what the structure of the TV used to be before you blasted it with a shotgun. And that's unfortunately more the case with collagen, because collagen isn't a crystal. And the minute you try and turn it crystal, it doesn't do what it did. All the things that are really interesting about it. I didn't know this when I didn't do this on my own. I was doing it with folk who were really into collagen. They just said, you just do this bit and don't ask questions. I didn't realise he was pushing the boundaries of what you could do with crystal. Oh, this is going on a hell of a tangent. Anyway, you need the right quality of X-ray. You can't just go to an X-ray machine and do that. You need a synchrotron, which we don't have very many of, because they're very expensive. And the one that we used was in Grenoble, which is shared across about 7 European nations, because it's too damn expensive. And it's a huge site. If you ever go to Google Maps and look at Grenoble from on high, you see a great big ring, which is a particle accelerator that takes the beams that have come out of the nuclear power plant they have sitting in the middle of it, which I wasn't aware of. So I went to stay there, and I was looking out the window of my accommodation and go, that's a strange looking domed building, that looks awfully like a nuclear reactor. And in fact it was.
What's it called again?
Grenoble.
No, the...
Oh, synchrotron, X-rays for the synchrotron. We have one in the UK called Diamond. Diamond is in the UK, but the one in Grenoble is this sort of European one.
I know what used to live there and I never came across it.
Seriously?
Yeah.
Please have a look at Google Maps. The thing is, it's massive. I mean, the site's a few square miles, at least a couple of square miles. You know, you have this massive facility, a nuclear reactor, great big long 400 metre something building where the beams come out, literally hole in the side of the nuclear reactor, let the beam come out, focus it, and then it goes into a particle accelerator for doing all the physics experiments. And what has this got to do with fruit flies? Right, you have this massive site. There are folk in there doing biology trying to figure out the structure of muscle. In fact, they decided they wanted to figure out the structure of fruit fly wing muscle. So what they had was a fruit fly in the path of the beam. There's this huge hall And they're sitting there with this poor, tiny creature. Unfortunately, they got slightly misaligned to start with and weren't going through its wing muscle at all. I mean, how the heck do you spot a wing muscle of a fruit fly, for goodness sake? Anyway, they were going straight through its head and it wasn't flapping its wings anymore as a result of that. I think they went through quite a few fruit flies. They should do it to midges, shouldn't they? I mean, let's do something useful with midges. I can't think. Anyway, I just thought it was interesting. And that's some of the stuff we've done with GAs, and it works. We were able to get a pretty good match of what the actual structure was by artificially evolving collagen to fit the real data. Sorry, was that? I don't know, it just amazed me that you have this massive site. It's amazing science, it's real science, I really enjoyed that. Anyway, I've gone off on a massive tangent, but it's related because I was using a GA to evolve stuff, and funnily enough, the next thing I would like to talk about is using a GA. So, you're familiar with GAs. How many here are not familiar with GAs? So you're not doing the optimization stuff. Okay. I will whistle stop through a GA. I think they're fairly effective. They're a part of a family of evolutionary algorithms. You don't just use GAs. There's lots of variants of it. There's things that take a principle and work with it. But the idea here is you've got some stochastic straight random process that you're running, and you would like to check that this process is reliable. You don't just take the first result you get, you run it a number of times and check that the result holds. This is a very common thing to do. The example I'm going to show you is you might want to know how mutation rate affects your optimization performance. Yeah, it has a massive effect. So you would run, say, 100 runs with one mutation rate, 100 runs with another mutation rate, and 100 runs with another mutation rate, and then have a look at the difference so you can figure out which one is going to help you solve your particular problem. What's the mutation rate you've been using? It's the rate at which you decide to take any individual offspring and just randomly change the values. So if you had a mutation rate of 0.5, you'd say half the time after I've got a new offspring, I'm just going to mutate some value of it. And that's, pardon? You mutate every single time. That's not good. If it's a GA, that's a mutation rate of 1. You always mutate. You always mutate your offspring. Oh boy. Is the GA doing badly? Yeah. Funny that. That's like, in reality, every time someone's born, they're always mutated. Well, that's actually not far off the truth. Okay, well, you'll be interested in the results of this study then, won't you? Right, so what do we do in a GA? Okay. The thing with GAs is, and this is the same with all of these heuristic algorithms, is you come up with some method of exploring your current solution space and some method of exploiting your current solution space. Have you heard this exploration? Yeah. And the exploring bit, which do you think does the exploring? If you've got breeding and mutation, which is the explorer?
Mutation.
Votes for mutation as the explorer, and votes for breeding as the explorer. Okay, the other thing you do is exploit what is working. So votes for exploitation being breeding, and votes for exploitation being done via mutation. Okay, the answer, I can't remember who said what, so you're good, is that mutation is about exploration. Yeah? Exploration is just trying something new. It's something my wife wishes I did a bit less of when we go hill walking, etc. Yeah? Breeding is exploitation. Yeah, it should be if you do it right. But if you get breeding wrong, you end up just doing exploration as well. If you just randomly mix stuff up, then you're just randomly mixing things. You're exploring. You go, I don't know where this goes, but let's just try it.
The thing is with the TSP, that's not a problem. There's no good crossover, so there's always a bit of...
There is, but you have to be very, very careful with it. You have to take big chunks. You can't take little pieces. The smaller pieces you mix and match, the more it just turns into random.
But I mean, if you take, so if you swap over a section,
Mhm.
in a large section, and obviously you're splitting that. But there's always a bit of shuffling to try and get rid of
Duplicates, yeah.
So there's always a bit of
Yeah.
exploration needed.
Absolutely. You've got to be really, really careful, and it is easy to trip yourself up. I'm glad you understand it, because I've been doing this particular problem for a few years now, and I keep trying to explain to my collaborator that this isn't quite as easy. To be fair, he's fairly convinced now, because I'll show you my results versus his. Anyway, so exploration versus, have you cleared that up? And you really don't want to be, well, if you're trying to go and get to the top of a hill, how much of that time do you want to spend randomly walking around the hill with your eyes closed? Very little. Yeah? If you've got mutation every cycle, that's what you're doing. You're going, I'm going to walk that direction. Oh, you know, I'll just go backwards for a minute. This is dangerous for me. I have got my eyes closed. Do you see what I mean? You, even if you think, oh, that looks good, and you start walking, but every time you go, I'll take one step that way, that's what you're doing by mutating every time. You're just randomly doing something different.
How do you know how to do one step?
Well, in the case of this, it's changing the sequence order of your visits and traveling salesman. It depends on the problem what a step means.
If the steps in order, if looking at an order rather than
Uh-huh. The step is making a change, but you don't know if it's a good change or a bad change until you make it. But in the case of the traveling salesman one, you are trying to maintain sequences. You don't. You're working on. I'm sort of jumping ahead of my slides, but what the heck. Now, can I go in the order of my slides, and then we'll come back to that. But you're all on the money, as it were. Right. So you probably figured out that making a GA go through the hoops is fairly simple. But would you agree with guiding the search process is less than straightforward? A bit of a dark art? Yes. The algorithm's really simple. But getting it to do what you want it to do and getting a crossover that works for your problem is actually quite hard. In fact, I've just found a paper which is written by loads and loads of people in the field noting about all the things GAs did to them to try and avoid solving the problem they'd set. Yeah? You have to be really careful about the question you ask it. It'll do a pretty good job of trying to solve it, but not by the route that you think. And we had that very thing in the results of the study we had, where we thought we were doing quite nicely, and then it went and made something worse. And then we looked at the question we'd asked it and realised, no, that's better than what we told it would have, but not what we meant. So you're familiar, you have a population of potential solutions, a population of routes, for example. You pick two parents based on the score. You don't pick the best every single time, because otherwise, yeah, if you pick the best every single time, what you're doing is try to hill climb. You're trying to just always, that's assuming that the problem is always steadily increasing, as in the solution always responds to a steadily increasing score, and taking two solutions with good scores and combining them will get you an even better score. That's not, on complex problems, that's not the case. Sometimes you have to get, well, your hill climbing is a good one. Your Scottish hill is a good one. You're at the top of a hill, well, you just keep following the gradient by picking the best parents, who are the ones that are highest up the hill. But actually what they're climbing is the false summit. And you've got another individual who's scoring not so well, but they've actually gone up the right they're going up the right slope. And if you just keep listening to your current best, they'll go better, 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 and they'll get to the top, and then they can't be improved on. And you've abandoned everybody else who was actually probably in the right place. Do you follow? You've got to be careful of not getting caught in what we call false minima. And if you keep picking the best parents every time, they're the locally best result you've got, but they may not be the globally best. So you've got to mix and match a bit. Yes, you sort of predominate towards the current best, but not to the exclusion of everyone. You need diversity.
If you randomly pick two terms from your population,
Yep.
and then you put all the tournament,
Okay, so the tournament selection is nice. So the idea with the tournament selection is you have a tournament of some size, say 5, so you pick 5 random individuals. You can use tournament selection to sort of very easily control the selection pressure. You you're picking the best of the tournament, not the best of the whole population. So if I have a population of 100 and I pick 5, there's only 5% chance that the best is in there.
That means the worst crossover they're going to get. Or they eventually.
Yeah, you're not likely, you're never going to get the worst one, but that's okay. You don't want to pick the worst one every time. That's not a way to solve a problem. But yeah, because if you've got a tournament of any size bigger than 1, then the worst one's always going to lose. That's good. That's what you want. What you don't want is the best always winning. But it's okay to have folk in the middle winning. You know, they don't look like the way forward at the moment. It turns out later on some of the material that was in there, some of the routes that were in there, would work for later. So what's lovely about the tournament selection is so easy to control selection pressure by just changing the size of the tournament. If you make the tournament 100% of the population, you have a hill climber. If you make it 95% of the population, then there's a 95% chance you're going to have the best in there every time. Yeah? So by just changing the tournament size relative to the population size, I can decide how much selection pressure I want. And in fact, most GAs, we vary it as time goes by. You can have very weak selection pressure to start with. You get into a solution and then you notice your scores aren't improving anymore. You widen it up again. You don't just stick with what you started with. You look at how well you're doing and you move things. And tournament selection is a really, really easy way to do that. The other easy way to change that, so that's you changing your exploitation pressure, and you can change your exploration pressure by just varying your mutation rate. If things have got a bit static, whack up the mutation rate and see if that puts you somewhere new. If that doesn't work, you might as well stop. Is this helping? I know it's not to do with D7, but it is what I do, so.
Yeah, this definitely helps. So I'm just confused with one single thing. When I'm doing a crossover and I have like 2 different, let's say like 13 A strings, I take like a chunk of the one and I like combine them, then I actually change a lot within the sequence.
Yep.
So it's just like more like a reputation move. When if I'm doing a mutation, I'm just like, for example, like picking 2 slots in that and invert that,
Yep.
I'm actually only changing like the difference of 2 single slots because the middle stays the same. Because to me it feels like the mutation is a smaller move along the gradient of the function, whilst the crossover can take me, let's say, like if the function is like this and I'm mutating only a little bit, it seems to me like climbing along the hill.
Yep.
But then if I'm crossing over and I have 2 very different chunks, this might mean that I'm jumping from here to here.
Yep.
So for me, what is like a little bit confusing right now.
It really can be. If you get, it very much depends on the problem, and it also very much depends upon how you decided to do crossover, whether crossover is crossover or it's just a variant of the mutation function. I've seen quite a few papers that annoyed me where folk have written off crossover saying you might as well just use mutation and be done with it, because the crossover function they've decided to use is, as you say, the equivalent of just mutating. So in the case of what you're doing, you're trying to preserve order. So I know that the thing with your mutation is it's just arbitrary about which ones you switch around. But if you're just crossing over, say, the first half of parent, well, some arbitrary length of parent 1 with the rest of parent 2 and then fixing it, that's less destructive because you're preserving quite a long sequence. And it is the case, you've got to take the thing that gets people is this is a probabilistic process, and yes, a lot of the time things are going to go wrong, but we don't care, because we just want, occasionally you hit the money, yeah? You're exploring and you're moving it out, and it just takes one of them to get it. The other thing, I'll come to your questions in a minute, the other thing I find people get confused about with evolution algorithms is they assume that it's a steadily increasing improvement. Do you see that? You don't, do you? You see improvement, and then it just plateaus off for a bit. Shouldn't go down. Yeah, and then it'll sit there for a while, and then it'll suddenly jump. And then it might improve, and then it'll level off. And that's what really should happen. Anyway, right, so your question.
Just going to say, ideologies like meiosis versus single nuclear informal office. So when you take things, you change something small. What we do in meiosis, which is the process of preparing the experiment X, you take big chunks of the games.
But you take order, you preserve order. Some problems you don't need to do that. Some problems each of the things in your chromosome, as it were, are independent. They're just parameters specifying something else. It doesn't matter, and you can just do this uniform crossover where you just take either parent A or parent B in each case. But in sequences, you can't do that. In things like Travis Salesman, that's highly destructive. I mean, if it helps any, GAs are the current best solution for the travelling salesman problem for I think over 6 million cities. Below that, there are better ways to do it, definitely. Linear programming works extremely well and absolutely hammers into this and is much faster. But it turns out, if you go beyond 6 million, then GA starts to win. But in fact, what they've done is they nicked a lot of the ideas from the linear programmers about what the linear programming was doing to make sure that the crossover was the right type of crossover. This isn't, what you're doing with your crossover is deciding how to take sequences across, and it's not as simple as perhaps the way you're doing it.
If you've got, say you've got a string on a certain length and
Yeah.
you're doing a crossover in the middle, and you're doing random points in the middle, should the length between those random points be a certain amount percentage of the string? Or
It depends on the problem, and no, I would say I can send you the paper with pieces. But yeah, well, I'll pick it out if you want to see it. It actually does a thing where it tries to preserve local roots in different parts of the graph, make sure that those local roots never get broken. So it doesn't try, it's quite careful about what it crosses over to make sure it doesn't end up just mutating every time. Anyway, I've gone off I can see we're going to be having a Monday lecture, aren't we? So on Monday I will finish off doing the GA stuff and show you how I've actually used it. And I have used it very, very effectively. It really works. And I can show you how well it works compared to a hill climbing approach, which my colleague tried first. I have a graph which basically changed the direction of our research. So I'll see you then. But we will not have a 5 o'clock lecture on Monday, I think.